Hello everyone. Welcome to Enzyme MBBS, where complex medicine becomes simple and exam ready. Okay, so without wasting any time, let's get started. So, coming to the definition, plasmids are extra chromosomal, double stranded, circular DNA molecules present in the cytoplasm of bacteria capable of independent replication. So, it's a very straightforward, easy definition. Just read once or twice, you will be able to write it. So, they are not essential for bacterial survival but confer selective advantages. We'll see what are the advantages. Coming to the key characteristics, again, whatever you written in the definition, just write it point wise separately and add some few extra points. So they are extra chromosomal genetic material, usually circular, double stranded DNA, located freely in cytoplasm, found mainly in bacteria because they are rarely found in yeasts, and replicate independently of bacterial chromosome because they have their own ori, that is origin of replication, so they are able to replicate independently. And size varies from 1 to 200 kb or kilobase. Now kilobase is something like 1 kilobase is equal to 1000 base pairs, so 200 is 200,000 base pairs, so it varies in that range. And number per cell is single or multiple means it would be up to 40 or more. And then coming to the plasmids versus episomes. Now, plasmids remain free in cytoplasm. Episomes are basically the plasmids that can integrate into bacterial chromosomal DNA. Just before we studied, plasmids are extra chromosomal DNA. Now, if they combine with the bacterial chromosomal DNA, they are known as episomes. Just draw that diagram too. Next, coming to the function of plasmids, we in the definition part we spoke that they are not essential but confer selective advantages. What are the advantages? Let's see. So plasmids carry genes responsible for, one is antibiotic resistance and virulence and toxigenicity, bacteriocin production. What is bacteriocin? Bacteriocin are antibiotic like protein substances that can kill other bacteria. Like if a bacteria produces the bacteriocin, it can use it to kill other bacteria, so they are bacteriocin. So that production and metabolic activities and conjugation. Then coming to the transmission of plasmids, how a plasmid can be transferred from one bacteria to another. So one thing is vertical transmission, it is to the daughter cells during binary fission it can be transmitted. And if we speak about the horizontal transmission, one is conjugation, it is during bacterial intercourse we can say, and one more is transduction. This happens via bacteriophages. Like bacteriophages are basically the virus which infect the bacteria, so if it goes and infect one bacteria, it takes that plasmid and it goes to another bacteria and it gives it there. So that kind also horizontal transmission takes place. Next, coming to the classification of plasmids. So here you can classify it based on three types. One is based on ability to perform conjugation. So it can be conjugative plasmid or non-conjugative plasmid. Conjugative plasmid basically have TRA genes, so they can transfer themselves. Example is F plasmid or fertility plasmid. The non-conjugative plasmids basically lack TRA genes, so they cannot self-transfer. Second type is based on compatibility. They are compatible plasmid and incompatible plasmid. Compatible plasmid, they basically different plasmid can coexist in same bacterial cell, whereas incompatible plasmid cannot coexist because both of them have same replication control. So basically they compete and eventually one will be lost and one will be survived. Next, coming to the based on function. Here, based on function, first thing is fertility plasmid or F plasmid. Again, we discussed this in the conjugative plasmid. They basically possess TRA genes and they form sex pili. Now, pili is the organ which helps in the conjugation. So, it enable bacterial conjugation by forming conjugation tube. So, example is fertility plasmid of E. coli. Next, resistance plasmids. As the name suggests, they carry genes for antibiotic resistance. Now, resistance may be due to penicillin, tetracycline, chloramphenicol, aminoglycosides, etc. Now, major cause of multi-drug resistance. Coming to the coal plasmids, they encode bacteriocins. We just few minutes before we spoke about bacteriocins, right? So, these are antibiotic like protein substances that can kill other bacteria. Then, virulence plasmid. So, they encode toxins and virulence factors. Example, heat labile and heat stable toxins by E. coli and siderophore production. What is siderophore? Siderophore basically helps the bacteria to get the iron. because in humans the iron is mostly in bounded form, it is not that free, so it cannot obtain that easily. So they produce something called as siderophore, which binds to the Fe3 plus and helps the bacteria to get iron. And next thing is adherence antigen, it helps the bacteria to adhere. So example is K88 plasmid in E. coli. Then metabolic plasmids, they enable bacteria to perform specific metabolic functions, for example digestion of unusual substances like toluene, camphor, etc., and urease synthesis. So urease basically helps in the production of ammonia, so they help the bacteria to tackle the pH environment, like acidic environment. So they use this urease, produce ammonia, and then they make it alkaline, so that helps them like that. And also the nitrogen fixation. Then curing of plasmids. What is curing? Curing is basically elimination of plasmids from bacteria. How we do it? Methods is, one is spontaneous loss, it can occur spontaneously, or it can occur by inhibiting plasmid replication. How we inhibit plasmid replication? There is various methods, like one is chemical agents, for example using acridine dyes, or else radiation, thymine starvation, growth at high temperature. All this basically inhibit plasmid replication. Last, plasmid as vectors, which is used in genetic engineering, so acts as vehicles to transfer genes. So widely used in recombinant DNA technology. I'm sure you would have studied about this in your previous classes. So genes inserted at specific restriction site, you basically use restriction enzyme, cut the gene of interest and plasmid it the same with the same restriction endonuclease, and you join that, form a recombinant DNA, then you transform. So all this process is very much familiar for you. So applications are protein production, example is insulin, gene therapy, vaccine development. So that's all about the plasmids. I hope the video was helpful for you. So if so, please like and comment, and keep watching. Do well, study well. All the best. Thank you.